this lecture, we are going to discuss about various DNA sequencing methods, that is the strategy to sequence nucleic acids, DNA or RNA. Now, the specific degradation and fractionation of the polynucleotide of interest to fragment small enough to be fully sequenced, that is the first requirement of DNA sequencing, that you could degrade very big fragments into smaller fragments and then sequence them. Another important point in basic strategy is the sequencing of the individual fragments. First thing is to fragment the DNA sample, that is larger DNA, then sequencing of that material, and then ordering of the various sequences of the fragments that is obtained to arrange or to yield the polynucleotide sequence actually through overlap regions. So, these are the basic 3 points about designing or planning a particular nucleic acid sequencing. Now, little bit in history, the first nucleic acid sequence it was obtained in almost like 65 by Robert Hawley, and it was 76 nucleotide molecule of yeast alanine transfer RNA. Some 12 years after Frederick Sanger had determined the amino acid sequence of insulin, like there was sequencing of numerous other species of tRNA and say 5S ribosomal RNA from several organisms was done. So, the art of RNA sequencing reached like in its zenith in 1976 with the sequencing by Walter Fires of the entire 3569 nucleotide genome of the bacteriophage MS2. Now, in contrast to RNA sequencing, DNA sequencing was in a far more primitive state because of the lack of available DNA endonucleases with any sequence specificity. Now, the first complete genome sequence which was determined was of the gram negative bacterium Haemophilus influenzae, which was reported in around 95 actually by Craig Venter. By 2010, the genome sequences of over 1000 prokaryotes had been reported, and many more is being determined now, as well as sequences of over 120 eukaryotes and many more in progress were reported, like those of humans also, and many other vertebrates, insects, worms, plants and fungi. A dramatic progress has been made in nucleic acid sequencing methods after 75, and there were 3 advances made this possible actually. One is discovery of restriction endonucleases to enable the cleavage of DNA at specific sequences. Then the development of molecular cloning techniques to permit the acquisition of almost any identifiable DNA segment in the amounts required for sequencing. Then the development of DNA sequencing techniques, various methods which have been developed, which we are going to discuss in here. Now, first method which was developed was chemical cleavage method, and it was developed by Maxam and Gilbert, that is Alan Maxam and Walter Gilbert, and it involved following steps actually, which is like one first thing was to radioactively label one end of the DNA, and that is the first step in the chemical cleavage method, to radio label one end, and usually it is used to be the 5 prime end, and it was labeled with the 32p. Now, if DNA already has 5 prime phosphate group, then it has to be removed by the alkaline phosphatase treatment, and then 5 prime terminus is labeled in a reaction with gamma 32p, ATP catalyzed by polynucleotide kinase. So, through this particular reaction, the 32p or the radio label p is incorporated. Then second step was to cleave the DNA in a base specific manner. Now, basic strategy for this chemical cleavage method is to specifically cleave the end labeled DNA at only one type of nucleotide. So, under conditions that each molecule is broken at an average of one randomly located susceptible bond. So, in this only the cleavage occurs at each base or a particular base according to the method or according to the strategy involved. Now, what will happen? This will produce a set of radioactive fragments whose members extend from the 32p labeled end, that is which is labeled from one end to one of the positions occupied by the chosen base where the cleavage has occurred. Now, always labeling can also be done on 3 prime end. So, both strategies could be adopted. Now, for example, if the DNA has to be sequenced, if you can see on your screen, if this is the DNA which needs to be sequenced, then it is labeled at 5 prime end with 32p. Always remember, left hand side is 5 prime and 3 prime is the left hand right hand side, unless it is specified. So, what will happen? Cleavage on the 5 prime side of the say for example, we can take one example G residue, then you will get following fragments, because like one cleavage will be occur after T and before G, that is at 5 prime side of G, and you will get this particular different fragments you are going to get here, which is smallest one will be T, because if it is occurring at this one, if it occurs at this G, then you will get another bigger fragment, another G, you will get this, and finally, when it is here, then you will get this fragment. That is what you can see on the gel when you are running the acrylamide gel. You can see only those which are labeled fragments. The other fragments which are gone will not be able to be seen here. Now, polyacrylamide gel electrophoresis will separate these fragments according to size, and remember, as we have discussed earlier, this can even differentiate between a single nucleotide. So, the position of the G residue in the DNA may be identified from the relative positions on the gel of their corresponding 32p labeled fragments as revealed by autoradiography. Now, in order for this method to work, the gel must be sufficiently resolving power, have sufficient resolving power to separate unambiguously fragments that differ in length by only one nucleotide, and as I said, it is possible in polyacrylamide gel. So, the DNA to be sequenced may be cleaved at a specific basis by subjecting it in a separate allocates, that is 4 different treatments are done, and then those fragments 4 reactions are run on the gel. So, here if you see if you go for each base, in ideally it should be A, T, G, C, but practically what is done is first there is a reaction for cleaving at G only. So, what is done is DNA is treated with dimethyl sulfate and which methylates G residue at N7 and rendering the glycosidic bond of the methylated residue susceptible to hydrolysis, and subsequent treatment by piperidine cleaves the polynucleotide chain before the depurinated residue. Then second reaction will be for A plus G, that is either A or G. So, the treatment with DMSF or dimethyl sulfate will preferentially methylate A residue at only N3 rather than N7, and so above treatment cleaves the DNA at A residue at only about 1/5 the rate it does at G residue. Now, but if this whole reaction is treated with acid, then both A and G are released. Remember, without acid only G is released frequently, but in acidic treatment both A and G will be released at comparable rate. So, that is another reaction, and then piperidine treatment causes strand cleavage before both A and G, and A residues are identified by comparing the position of G and the A plus G cleavage. The third reaction could be C plus T. Now, the reaction of DNA with hydrogen followed by piperidine treatment cleaves DNA before both its C and T residues. And then fourth reaction alicate will be for only cleavage at C. Now, here DNA is treated with hydrogen in salt actually 1.5 molar NaCl, and where cleavage will occur only at C residue. This way you can compare C and C plus T and then find out which residue it is. Now, in all 4 reactions the conditions are adjusted so that the strands are cleaved at an average of 1 randomly located position each. Now, cleavage fragments which are obtained, they will be separated on the basis of size. So, the 4 differently fragmented sample of DNA, that is A plus G, G, C and C plus T will be simultaneously electrophoresed, and then it is a sequencing gel, as we have discussed earlier. It is done at very high temperature and 8 molar urea to avoid any base pairing and ensure that DNA fragments are separated only according to their size. And this could be read directly of an autoradiogram of the sequencing gel. So, if you can see on your screen, this is a basic strategy here is that first thing is you convert it to single stranded DNA, mostly here single stranded DNA is done in Maxim and Gilbert method. Then it is labeled at 5 prime end and 4 tubes that contains 4 reaction, that is to cleave at base G or A or G, T and C and base C will be performed as we have discussed, and then finally it will be run on the electrophoresis. So, if you can see these are like the lanes for each of the reactions, and if you can see here this particular one, this was the sequence, and as we have gone through you can see the sequence could be built by starting from the bottom. Now, remember when the cleavage will occur at 5 prime side of this G here, earlier we have taken the sequence which was T, which was different little bit, it was TGT, but just to understand that if it is like here, then there will be only labeled P actually and no base. So, that will sometimes is difficult to see in here. So, that way the whole sequence can be generated except for first base actually. Now, this was Maxim and Gilbert method, and it was preferred method at that time for sequencing, and but it is no longer used actually. After that the Sanger method was very popular, and it is called chain termination method or dideoxy chain termination method. What it does is it utilizes the clino fragment of DNA polymerase to synthesize complementary copies of the single stranded DNA being sequenced, and it lacks 5 prime to 3 prime exonuclease activity. Now, DNA to be sequenced is incubated with the clino fragment, a suitable primer like here PCR is required, like as we have discussed in earlier lecture, and 4 DNTPs. Now, here what happens is that either at least one DNTP or the primer is labeled here. So, that this could be read actually. So, either primer could be labeled or DNTPs is labeled. So, what is done here, like if this is primer and this is template, as it extend across, then a new strand will be synthesized on this primer. So, in this case what is done is, in this method a small amount of 2 prime 3 prime dideoxynucleoside triphosphate is added to the reaction mixture. So, what will happen? So, these are like if you see on your screen, this is normal DNTP, and this is where you have 2 H here at both 2 prime and 3 prime side. So, the extension will not occur, the next reaction will not occur actually in chain extension. So, when the dideoxy analog is incorporated in the growing polynucleotide in place of the corresponding normal nucleotide chain, growth is terminated because of the absence of 3 prime OH group. So, by using only a small amount of dideoxy nucleotide, a series of truncated chains are generated, and each of which is terminated by a dideoxy analog at one of the positions occupied by the corresponding base. Now, each of the 4 DD NTPs is reacted in a separate vessel. Now, the 4 reaction mixtures are simultaneously electrophoresed like it was done in Maxim and Gilbert method in parallel lines on a sequencing gel, and this is a long thin gel and it contains like earlier 7 molar urea and run at 70 degrees Celsius to avoid the hydrogen bonding and ensure that DNA fragments separate only according to their size. The sequence of the DNA that is complementary to the template DNA can then be directly read off an autoradiogram of the sequencing gel from bottom to top, like it was done in Maxim and Gilbert method. If you see, the smallest one was at the bottom, and then the largest fragment which is like you go in that particular sequence. However, a single gel is incapable of resolving much more than 300 to 400 consecutive fragments. The limitation is you can have you can be generated like you can run more than 2 sets of gels, one run for a longer time and higher voltage than the other to obtain the sequences up to say 800 base pair DNA fragments. But that is a different strategies, like one can fragment and many fragments of 300 to 400 base pairs could be run, or it could be other way that you can have longer one. The absolute requirement is that DNA to be sequenced is in a single stranded form, as it is like it could be run on the gel, and to facilitate the isolation of single strands, DNA to be sequenced may be cloned into one of the clustered cloning sites in the LEC region of M13 MP series of vectors. A feature of these vectors is that cloning into the same region can be mediated by any one of a large selection of restriction enzymes, but it still permits the use of single sequencing primer. There could be many modifications of chain termination sequencing method. One, the sharpness of the autoradiographic image can be improved by replacing the 32p radio label with a much lower energy like say 33p or 35s. In the case of 35s, this is achieved by including an alpha 35s deoxynucleoside triphosphate in the sequencing reaction, and this modified nucleotide is accepted by DNA polymerase. So, there is no problem about that, and it is incorporated into the growing DNA chain. Now, non-isotopic detection methods have also been developed with chemiluminescent chromogenic or fluorogenic reporter systems. Now, although the sensitivity of these methods will not be as great as with radio labels, but they are adequate for many different purpose. You can see here this 35s incorporated in here at alpha position. There are other technical improvements to Sanger's original method have been made by replacing say clino fragment of E. coli DNA polymerase 1, natural or modified forms of the phase T7 DNA polymerase have found favor, as has the DNA polymerase of the thermophilic bacterium like Taq polymerase, and other polymerases have also come in picture. The T7 DNA polymerase is more processive than clino polymerase, that is, it is capable of polymerizing a longer run of nucleotide before releasing them from the template. Its incorporation of dideoxynucleotides is less affected by local nucleotide sequences, and so the sequencing ladder comprises a series of bands with more even intensities. Taq DNA polymerase can be used in chain termination reactions carried out at high temperatures, and this minimizes chain termination artifacts caused by secondary structures in the DNA. Then Tabor and Richardson in 95 shown that replacing a single phenylalanine residue of Taq DNA polymerase with a tyrosine residue results in a thermostable sequencing enzyme that is no longer discriminates between dideoxy and deoxynucleotides. Combination of chain terminator sequencing and M13 vectors to produce single stranded DNA is very powerful. Very good quality sequencing is obtained with this technique, especially when the improvements given by 35S labeled precursors and T7 DNA polymerases are exploited. So, there were further modifications have been done which allow sequencing of double stranded DNA. Double stranded input DNA is denatured by alkali and neutralized, and then one strand is annealed with a specific primer for the actual chain terminator sequencing reaction. This approach has gained in popularity as the convenience of having a universal primer has grown less important with the widespread availability of oligonucleotide synthesizers. So, with this development, Sanger sequencing has been liberated from its attachment to M13 cloning system. For example, polymerase chain reaction amplified DNA sequencing can be sequenced directly also actually. One variant of the double stranded approach often employed in automated sequencing is cycle sequencing. This involves a linear amplification of sequencing reaction using say 25 cycles of denaturation annealing of a specific primer to one strand only and extension in the presence of tag DNA polymerase plus labeled dideoxynucleotides. Now, alternatively labeled primers can be used with unlabeled dideoxynucleoside. So, here this schematic shows this particular method here. So, what you have is templates and a primer which is on one side. So, primer will this will be extended by the polymerase, and what you get is according to the particular dideoxy nucleotide you will get these fragments here, because they will be like it will be interrupted at different forms, different bases actually. So, if you have DDATP say, then it will be like A will be these fragments will be generated as complementary to this template fragment. Likewise, for G for C and for G and for dideoxy T. So, that way, and if you run on these fragments, then you will get different sized fragments which are terminated fragments, and as you read it from bottom to top, like you can read the you can make out this particular sequence actually, which is CCGG CC8TC from 5 prime to 3 prime. So, likewise, this is like as you have seen here, this is GG. So, it will come CC here, then A, it is 8, then T, then CC, CG, and likewise you will get the sequence actually. So, reading from top to bottom excluding the primer, you will be able to read the sequence from the sequencing gel on autoradiogram. Now, sequencing methods, DNA sequencing methods have been automated and a lot of manual intervention is lessened actually. So, in order to sequence very large tracts of DNA such as entire chromosome, the Sanger method has been greatly accelerated through automation actually. This required that the above described radio labeling techniques, which are only which are not readily automated, be replaced by say fluorescence labeling techniques actually. So, it has benefit of eliminating the health hazards and storage problems of using radio labeled nucleotides. In the most widely used such technique, each of the 4 DD-NTPs used to terminate chain extension is covalently linked to a different fluorescing dye, that is with different colors, and the chain extension reactions are carried out in a single vessel actually containing all 4 of these labeled DD-NTPs. So, it is not separately done, it is not 4 reactions, but since you can see the colors, that is 4 different dyes, it is done in the same vessel. The resulting fragment mixture is subjected to sequencing gel electrophoresis in a single lane. Now, as each fragment exits the gel, its terminal base is identified according to its characteristic fluorescence spectrum by a laser activated fluorescence detection system. The fluorescence detectors used in these devices, which have error rates of say 1%, are computer controlled, and hence data acquisition is automated. Now, in most of the advanced systems, such systems actually, the sequencing gel is contained in an array of say up to 96 capillary tubes rather than in a slab shaped apparatus. Sample preparation and loading are performed by robotic systems, and electrophoresis and data analysis are fully automated. For high sensitivity DNA detection in 4 color sequencing and high accuracy based calling, one would ideally like the certain criteria to be met. One is each of the 4 dyes to be exhibit strong absorption at a common laser wavelength. So, that is important that all the 4 dyes are detected at same level, to have an emission maximum at a distinctly different wavelengths actually, that they emit at distinctly different wavelengths, and to introduce the same relative mobility shift of the DNA sequencing fragments. And now, recently dyes with these properties have been identified and successfully applied to automated system actually. There are a lot of advantages of automated DNA sequencing method. One is that manual sequencing can generate excellent data, but even the best sequences in laboratories due to like say poor autoradiographs are frequently encountered, and that will make sequence readout difficult or impossible. Usually the problem is related to the need to run different termination reactions in different tracks of the gels. Even skilled DNA sequencers ignore bad sequencing tracks, but many laboratories do not. Now, this leads to poor quality sequencing data, and the use of single gel track for all 4 dideoxy reactions means that this problem is less acute in automated. So, one is that many times you might get poor data due to certain practical problems. Uh, so, uh, in automated sequencing this will be little less. Nevertheless, it is desirable to sequence a piece of DNA several times. One should do it to confirm the sequence, and many times the 2 strands are also sequenced to confirm the obtained sequence is right. And this eliminates errors quite a lot actually. It should be noted that long runs of the same nucleotide or a high GC content can cause compression of the bands on a gel. If there is a same nucleotide, a long stretch of that or high GC content, then it could be a problem necessitating manual reading of the data. Even with an automated system, this could be a problem. Note also that multiple tandem short repeats, which are common in the DNA of higher eukaryotes, can reduce the fidelity of the DNA copying, particularly with Taq DNA polymerase. Second advantage of automated DNA sequencing is that the output from them is in machine readable form. So, this eliminates the errors that arise when DNA sequences are read and transcribed manually. In the new generation of the slab gel, like in new generation of sequence, slab gel is replaced with like I said 48 or 96 capillaries filled with the gel matrix. The equipment has been designed for use with robotics, therefore, minimizing hands on time and increasing the throughput. With a 96 capillary sequencer, you can sequence very long fragments per day, actually many nucleotides per day. So, on your screen if you can see here, when you take 4 fluorescent dyes attached, different colored dyes are attached to each base or each dideoxy analogues, then what you get fragments here as they are of different sizes as the PCR goes on, and these fragments you can see when they run on a gel, on the same gel, and they are in the same, like extensions are done in the same vessel actually. So, what you get is uh, uh, as they run, you can see that colors could be monitored through the laser here and data could be recorded, and based on the color of the dye, the base could be identified, and you will get a chromatogram in this form, where you will get different colors as they move out of the base of the gel, and they could be a proper and very accurate sequence could be obtained. Now, genome sequencing is one another challenge actually. So, like we have seen in Sanger's method, fragments could be done, and certain DNA fragments could be sequenced without any problem, that you can fragment them and you can sequence those fragments and overlap them on a smaller scale. But when you come at a genome level, there is a major technical challenge in sequencing a genome. Uh, in the and is like when you are sequencing a genome, there the DNA sequencing is like assembling the tens of thousands to tens of millions of sequenced segments here, depending on the size of the genome. So, it is not like one DNA or a few DNA fragments, it is a thousands of DNA fragments has to be arranged into contiguous blocks actually, or called contigs, and assigning them to the proper position in the genome. So, it is a much more complex task, much more challenging task than sequencing a longer DNA fragment. Now, one way that contigs might be ordered is through chromosome walking actually. However, to do so for eukaryotic genome would be prohibitively time consuming and expensive. So, there are certain techniques which have been developed for genome sequencing, and as you heard about human genome and other eukaryotic and other genomes have come up, they have been done. The two strategies has been employed. One was map based genome sequencing. So, this is efficient technique of genome sequencing, and it was developed in the late 80s. It is a role. So, what is done, low resolution physical maps of each chromosome are prepared by identifying shared landmarks on overlapping say 250 kilobase insert that are cloned in each artificial chromosomes. Now, these landmarks often take the form of 200 to 300 base pair segments, which are known as sequence tagged sites or STS, and whose exact sequence occurs nowhere else in the genome. Hence, two clones that contain the same STS must overlap. Now, the STS containing inserts are then randomly fragmented into say 40 kb segments that are subcloned into cosmate vectors, so that a high resolution map can be constructed by identifying their landmarks overlaps. Now, the cosmate inserts are then randomly fragmented into overlapping 5 to 10 kilobase or 1 kb segments for insertion into plasmid or say M13 vectors. Now, these inserts, which could be like say 800 M13 clones per cosmate, are then sequenced like 400 base pairs per clone, and the resulting so called reads are assembled computationally into contigs to yield the sequence of the parent cosmate insert, with the redundancy of say 400 base pairs per clone into 800 clones per cosmate. It could be you can get redundancy of say 8, which is you have 40,000 base pairs per cosmate. So, it could be like you can get redundancy of around 8 actually. Finally, the cosmate inserts are assembled through cosmate walking using their landmark overlaps, and these landmarks ideally spaced at intervals of say 100 kilobase or less, and you will sequence of the each artificial chromosome inserts, which are then assembled using their STS to yield the chromosomes sequences. Now, genome of most of the complex eukaryotes contain numerous tracts of repetitive sequences. and such repetitive sequences greatly exacerbate the difficulty of finding properly spaced STS. To partially circumvent the latter difficulty, STS like sequences of cDNAs, which are known as expressed sequence tags, are used in place of STS. Now, since the mRNAs from which cDNAs are obtained, which are reverse transcribed and they encode proteins, they are unlikely to contain repetitive sequences. So, this is one method which is map based sequencing, and as you have seen it is like first is to clone it in certain fragments in yeast artificial chromosome, then cosmate, then certain vectors, and finally, those sequences has to be assembled actually. There is another approach that is called whole genome shotgun assembly strategy. The whole genome shotgun assembly strategy is formulated by Craig Venter, Hamilton, Smith, and Leroy Hood. And this strategy, in this strategy, a genome is randomly fragmented into a large number of cloned fragments. First, it is fragmented, and then large number of cloned fragments are then sequenced, and then genome is assembled by identifying the overlaps between pairs of fragments. So, here it is a random shotgun method adopts random fragmentation, cloning, and then sequencing, and then identifying the overlaps to arrange the sequences in a proper order. For bacterial genomes, this shotgun approach is carried out straightforwardly by sequencing tens of thousands of fragments and assembling them. Then in a task known as finishing, actually the gaps between contigs are filled in by several techniques, including synthesizing PCR primers complementary to the ends of the contigs and using them to isolate the missing segments. For eukaryotic genomes, there are much greater sizes are there and require strategy to be carried out in stages, actually this shotgun strategy which is carried out in stages. So, here a bacterial artificial chromosome library of say 150 kilobase inserts is generated. The insert in such of these artificial chromosome clones is identified by sequencing 500 base pair in from each of the end to yield segments known as sequence tagged connectors or BAC ends actually. Now, one BAC insert is then fragmented and shotgun cloned into plasmid or M13 vectors. The fragments are sequenced and assembled into contigs. Now, sequence of this seed BAC is then compared with the database of STCs to identify the 30 overlapping BAC, say up to 30 overlapping fragments here. 30 overlapping BAC clones. The two with minimal overlap at either end are then selected, sequenced, and the operation repeated until the entire chromosome is sequenced. This is called you can say BAC walking, actually bacterial artificial chromosome walking, which for the human genome required 27 million sequencing reads actually. So, these are two strategies here compared. In say map based strategies like we have discussed, there is a chromosome which is 250 million base pairs. There is like first artificial yeast artificial chromosome library is made, and then finally cosmate library is made, and then they are fragmented and cloned into M13 or plasmid library, which are then chosen to be sequenced, and then cosmate walking is performed, and finally the sequences are obtained and they are properly arranged. In another method where that is shotgun approach, where chromosome is taken and bacterial artificial chromosome library is prepared, then after that M13 or plasmid library is prepared, then lot of these clones are sequenced and assembled. Here it is randomly fragmented, and then these overlapping, this is like called BSC walking, and then this particular whole sequence is obtained. There are certain advantages of shotgun strategy over the map based strategy. The shotgun strategy is readily automated through robotics and hence is faster and less expensive than the map based strategy. Most known genome sequences have been determined using the shotgun approach, many in a matter of say few months, and its advent reduced the time to sequence the human genome also by several years. Nevertheless, it appears that for eukaryotic genomes, most of the residual errors in WGSA, or you can say shotgun based genome sequencing, can be eliminated by finishing it through the use of some of the techniques of the map based strategy. Now, if we just little bit discuss about human genome has been sequenced, which rough draft was reported in 2001 by two independent groups, publicly funded International Human Genome Sequence Consortium, which is like 6 countries were involved and were led by Francis Collins, Eric Landor and John Sulston, and which used the map based strategy, and then a privately funded group from Celera Genomics led by Craig Venter, which used the shotgun approach. So, the first one determined genome sequence was a conglomerate from numerous individuals, whereas that from Celera Genomics was derived from 5 individuals, but mainly from Craig Venter. Now, these draft sequences lacked 10% of the gene rich chromosomal regions known as euchromatin and much of the largely unexpressed chromosomal regions known as constitutive heterochromatin. Both draft assemblies had sequencing errors rates of say 1% and contained around 1 lakh gaps, so that 1,60,000 gaps around that, so that the order and the orientation of many contexts within the local regions had not been established. So, in 2004 the public funded consortium reported the finished sequence. It covered almost 99% of the euchromatic genome, and with a error rate of 0.001% and had only 281 gaps. In 2007, Venter reported the finished sequence of his own diploid genome, and these stunning so it is a very great achievement actually, and these stunning achievements, the culmination of over a decade of intense efforts by hundreds of scientists, has really revolutionized the way both biochemistry and medicine will be viewed and they will be practiced actually. Few of the major observations here were that about 40% of the human genome consists of repeating sequence of various lengths, and only 28% of the genome is transcribed to RNA. Then only 1.2% of the genome, that is 4% of the transcribed RNA, encodes protein, and it is said that human genome appears to contain around say 23,000 odd genes or protein encoding genes, which are open reading frames. Earlier it was thought that more than 50,000 open reading frames are present. Only a small fraction of human protein families is unique to vertebrates, and most occur in other life forms also. Two randomly selected human genomes differ on an average by only one nucleotide per 1000, and that is that two people, if we compare, they will have 99.9% genetical identity, or they will be identical actually. So, this is like only 0.1% makes so much of difference. Genomes of eukaryotes, including that of Homo sapiens, can be explored at this given website, and so this was really a great achievement, and many genomes are being sequenced and reported. Now, these were methods which were advanced, like map based or shotgun approach were advanced DNA sequencing method, but now there are next generation methods of DNA sequencing have come in, which is like second generation you can say, and third generation methods have come in. We will discuss some of them here. One is the 454 sequencing system actually. The Watson genome was sequenced using a system developed by 454 Life Sciences that employs the following methodology. The methodology employed is here that genomic DNA is randomly sheared to small, say 300 to 500 base pair fragments and ligated to adapters, which in turn are specifically bound by say 30 micrometer diameter DNA capture beads under dilution conditions, such that at most one DNA fragment is bound to each bead. Now, the beads are suspended in a PCR mixture containing DNTPs, primers complementary to the adapters and tagged DNA polymerase. The suspension is emulsified with oil such that each aqueous droplet contains only one bead, and that is each bead is contained in its own micro reactor, thus preventing the introduction of competing or contaminating sequences. The PCR is carried out by thermocycling until 10 million identical DNA fragments are bound to each DNA capture bead. The emulsion is broken by the addition of isopropanol. The DNA is denatured and the resulting single stranded DNA bearing beads are deposited into 5 picoliter wells on a fiber optic slide with one bead per well and the slide containing 1.6 million wells. So, this is how it is done. These are like fragments, there is a bead here on which each one fragment is processed, and these are wells where these beads are put in. The DNA on each of these beads is sequenced using a series of coupled enzymatic reactions, and these are collectively known as pyrosequencing. So, in pyrosequencing, a solution containing only one of the 4 DNTPs is flowed over the bead containing slide. Now, if that DNTP is complementary to the first unpaired base on a template strand, DNA polymerase catalyzes its addition to the primer strand and releases pyrophosphate ion. In a reaction catalyzed by the enzyme ATP sulfurylase, the pyrophosphate ion reacts with adenosine 5 prime phosphosulfate to yield ATP. Now, in a reaction catalyzed then by F enzyme luciferase, the ATP reacts with luciferin and O2 to yield oxyluciferin and a flash of visible light. The well from which the light flash emanates together with its intensity is recorded by an imaging system, and thus identifying those wells in which the foregoing nucleotide was added to the primer strand. The intensity of the light is proportional to the number of nucleotides reacted, so that when 2 or more consecutive nucleotides of the same type are added to the primer strand, their number is determined. In preparation for the next reaction cycle, any unreacted DNTPs and ATPs are hydrolyzed to mononucleotides and phosphate ion by wash containing the enzyme apyrase. So, these are the way, these are different reactions as we have discussed through various like first DNA polymerase, then ATP sulfurylase, and then finally luciferase where light is emitted, and then finally washing steps are performed in this particular method. Now, this series of reactions is automatically iterated by sequentially using all 4 DNTPs and then repeating the entire process. In this way, the sequence of say around 400,000 DNA fragments can be simultaneously determined to a length of 400 nucleotides, each with an accuracy of 99% in one say 4-hour run. So, hence the 454 system is over 300-fold faster than the Sanger method of sequencing. Then there are other DNA sequencing platforms are there, like first next generation instrument genome analyzer was developed by Solex Inc., which was later purchased by Illumina. It utilizes a strategy for massively parallel DNA sequencing, whereby individual DNA fragments with appropriate linkers are first amplified on a slide matrix similar to microscopic slide using a solid phase PCR process. The slide with those so called clusters of amplified fragments is then placed onto the genome analyzer, and the 4 bases are added one at a time to the entire slide in a repetitive cycle. Then genome analyzer utilizes fluorescently labeled terminators that allow detection of fluorescence by a sensitive camera of single base incorporation events into growing DNA strand. The terminator molecules are designed such that 2 sequential bases cannot be added in the same reagent additional cycle. There is another system called sequencing by oligonucleotide ligation and detection. This is from applied biosystem. Here this can simultaneously sequence say 180 million DNA fragments with read lengths up to say 50 nucleotide each for a total of 9 billion nucleotide in a single run. A library of DNA fragments is prepared here from the sample to be sequenced. Individual DNA fragments have sequencing primers ligated onto them and then are amplified using emulsion PCR on tiny beads. Beads with amplified DNA on them are then purified and covalently linked to a solid surface to a slide surface. Smaller bead size enable much higher number of simultaneous DNA sequences to be analyzed than the previous 2 platforms. The ABI chemistry utilizes sequencing by ligation. In this method, fluorescently labeled oligonucleotide probes are ligated to the primer only if they are perfectly matched to the upstream sequences. This ligated piece of DNA now serves as a primer, and the next labeled probe is ligated to this if it matches the upstream sequence. So, significantly higher specificity is obtained and higher accuracy also than the sequencing by the synthesis approach. So, this is how this reaction is carried out. This is like beads here on which it is attached and PCR is carried out, and this is the real reaction here where priming and ligation are 2 methods. So, what is done, if you can see on the screen, there is a adapter and there is a primer against that. Now, this fragment which is the probe you can say, and this is like what you call fluorescence material is in here. So, this will be matching and it will as per like it is matching after the primer it will sit in here, and these are various probes in here. So, one which matches here will be will complement, then this here gap is filled in by the ligase. So, ligation is done. After ligation this is excited and fluorescence could be obtained for this, and then this is removed here, unextended strands and other things will be removed. Second is the floor containing a few bases is cleaved off, and then this the steps from 1 to 4 are repeated. So, what you get, if you see here, there is 1 2 bases here which could be which are known or in the probe, then there are other 2 bases, then other 2 bases. Likewise, you will get sequence through priming and ligation, that is each time these fragments different types with containing 2 you can say there are 2 bases probes, die base probes you can say, and these are 6 to 8 base long, and when they are attached through ligation and finally they are removed here, fluorescence could be read, and then these are repeated these steps. Finally, primer reset is done where this whole thing is removed and the primer is taken which is n minus 1 base pairs of the original primer. Then again these steps are repeated and almost like 5 cycles are done with each time 1 nucleotide is removed, like n minus 1 n minus 2, likewise it will be done. This is summarized in here. If you can see, this is the original primer, then there is n minus 1 primer, n minus 2 primer, and if you can see in each cycle there are certain overlaps, like these 2 bases were the first one, these 2 bases here, and then next 2 bases were again these were the next 2 bases. Likewise, there is a overlap here of these sequences, and finally, you will get the full sequence as you go. Each time you ligation and priming reaction with one short nucleotide shortened primers, and different probes are performed. The second next generation instruments, which is single molecule sequencers, the dramatic output of DNA sequence achieved by the first next generation DNA sequencers was obtained by utilizing massively parallel DNA sequencing, and very small reaction volumes significantly reduced the reagent cost. But this was previously done. However, all of these platforms depend on PCR to create sufficient mass of the DNA fragments to be analyzed. Now, sequencing of these amplified individual templates causes two problems. One is PCR is relatively expensive and time consuming, and second is the amplification process is not perfect and can in rare cases introduce erroneous bases and the amplified products in amplified products, and these errors are perpetuated in the DNA sequences obtained from these amplified products and will increase the error rates. So, one solution to both problems is the development of sequencing platforms that can analyze individual single molecules of native DNA without the need for PCR amplification. So, this second next generation methods do that, and there are two methods we are going to discuss. One is heliscope single molecule sequencing. Now, the first single molecule sequencing machine was developed by Helicos. This platform uses individual DNA fragments that have been tailed with poly DA and then annealed to a slide containing a lawn of oligo DT primers. The slide is then sequentially exposed to each of the 4 bases and imaged to determine which DNA fragments have incorporated a specific nucleotide. This machine produces very short sequence reads, but on hundreds of millions of templates, yielding multiple gigabases of DNA sequences per run. There is another method called single molecule real time sequencing, and it is produced by Pacific Biosciences. This sequence platform tethers a single DNA polymerase at the bottom of an optical chamber known as zero mode waveguide. This waveguide is a structure that creates an illuminated observation volume that is small enough to observe by laser induced fluorescence a single nucleotide of DNA being incorporated by DNA polymerase. The 4 nucleotides are fluorescently labeled, and as they are incorporated by the DNA, they could be by the DNA polymerase, the cleavage of the fluorescent terminator by the polymerase can be detected prior to the next base being incorporated. Thus, this instrument can obtain sequence information at the processivity rate of DNA polymerase, which is several hundred bases per second. So, these are also called like second or third generation methods actually. It depends on how you call, which you call first generation, or you can call these are next generation platforms. So, this completes our lecture. I think one could look into details of the two methods we discussed in the end, but we have discussed in this lecture in detail about various methods like map based methods or shotgun approach. Likewise, we have discussed Sanger's dideoxy method and also Maxim Gilbert very old method. All these methods and new generation platforms has really enhanced the efficiency of sequencing, and the time has been reduced quite a lot for sequencing. This is helping in sequencing many complex genomes of eukaryotes as well as prokaryotes, and will certainly help in understanding many different questions which are raised regarding in this particular area of biotechnology, and these sequences will certainly answer them to satisfactory level. Thank you.